This is podcast number 31. In this podcast, we're going to talk about viruses and whether or not they can be considered alive. Like other podcasts from chapter 8, not going to give you a straight answer, and that's not because I'm trying to confuse you. It's just because there isn't a straight answer. So it depends on how you define life. And scientists disagree. Just like scientists may disagree on what is or is not a species, scientists might disagree on what is or is not a cell. So there's a lot of viral diversity out there, and viruses have a genome. The genome can be single-stranded DNA, double-stranded DNA, single-stranded RNA, double-stranded RNA. And the genome size can vary pretty wildly, from really small to fairly big, bigger than some well-defined cells, things that other people don't consider controversial at all, whether or not it's a cell. The SARS coronavirus, this is talking about COVID from 10 years ago, not the current one, but the current one is closely related to that and has a similar size genome. And the number of genes can be very small to very big. So how do you decide whether or not something is alive? You got to decide on what is this life? How do you define life? And that's really hard to do. Life is an emergent property, which is one of the big ideas of this book in this class. Is emergent properties emerge or come out of things that you can't dissect down and find. So if you take apart a cell into its component parts, its organelles, its membranes, and then maybe its proteins, nucleic acids, carbohydrates, and lipids, those things aren't individually alive. Maybe you could say an organelle is alive, like mitochondria could, quote unquote, live outside the cell for a while. But life emerges from the assembly of those things. And certainly we have uncontroversial ways of saying that a cell is alive or a cell is dead, or an organism is alive or an organism is dead. But what is it that comes together to make that happen? And what are we going to quantify as scientists when that does happen? That's hard to say. So here are some things that you could use to say whether or not something is alive. So you could say that it exists as one or more cells. It uses and synthesizes genetic information to reproduce. It descends from other beings and is the product of evolution. Produces molecules from environmental substrates. This is manipulating, transferring energy from one molecule to another. Harvests energy and uses it to perform functions. Energy in the form of chemical bonds, energy from the sun, energy from heat. And they regulate their internal environment. They have some kind of homeostasis, a way of regulating their environment. So, do viruses fit this definition? Well, let's look at HIV. HIV is the human immunodeficiency virus. It's the virus that causes AIDS. These little green things here are false color image of a scanning electron micrograph of viruses budding from an infected white blood cell. Human membranes form an envelope around the virus, so it's an enveloped virus, like we consider cells have a membrane-bound area. There is a protein coat called a capsid, and there are nucleic acids inside. So in this diagram, here's the envelope that is membrane, both protein and lipids. That may be studded with proteins. There's going to be a capsid. Sometimes there's not always a membrane around viruses, but there is for HIV. And there are proteins associated, and there's nucleic acid associated with it. If you look at the genome laid out from one end to the other, there are several genes in the genome. We know what those genes do, and it's small. So our genome is 3.1 billion base pairs. The HIV genome is less than 10,000 base pairs, and it can kill us. The COVID-19 virus is also an enveloped virus, has a membrane around it. It has a mRNA for its genome, unlike HIV, but it has RNA for its genome. It has single-stranded RNA, and this single-stranded RNA is delivered once it gets inside the cell and then read by ribosomes inside the cell to make the proteins to make more virus. It can use energy from the cell in order to do that, and it is evolving. We know that several coronavirus genomes exist. Hundreds of coronavirus genomes exist. Mutations have happened, so there's been a change in gene frequency in the population of coronavirus since it was first isolated in December until now, and so by definition it has evolved. Maybe you could use the size thing again, like we tried to talk about with cells, below a certain size. But just as with cells, with viruses, there are exceptions of viruses that are just enormous. So Mimivirus is a very large virus. Scale bar here, 100 nanometers. It's a very large virus on the order of this and bigger than some bacteria. It has this starfish shape, a five-fold sort of symmetry to it that will open up to release its viral genome. And when it invades its host is an amoeba, the viral genome takes advantage of the host genome and takes over to synthesize more copies of the virus, similar to lots of viruses. If you take the many genes that are in Mimivirus and then use the sequence of those genes to try to figure out, well, how is this thing related to other life that we know? So the three domains of life, eukaryotes, archaeans, and bacteria, where is Mimivirus? And it puts it on the eukaryote branch of the tree. So what about a definition that said, okay, well, viruses can infect cells, but no viruses can infect other viruses? That might be a useful definition. Well, that's not true. So about 10 years ago, there was a virus called Sputnik that was shown to infect this Mimivirus, this giant virus. So the poor amoeba has this giant virus inside of it, and then inside that virus is another virus. And they called that a virophage. Okay, so that's not going to work. And it actually reproduces inside another virus. So it's not just a fluke that it invades the other virus, it acts like a virus inside a virus. Are there other things that are not viruses but sort of act like viruses. There are. So there are proteins called prions that cause mad cow disease. Mad cow disease is a disease that got its name because cows can be infected with these infectious proteins, and then people who eat the flesh of these cows, even if it's been cooked, can also be infected. So this is transmitting from one organism to the next through protein, and there's no nucleic acid involved at all. So all the virus cases, all the infectious cell cases, like if you get infected with a bacterium or something like that, nucleic acid is involved in that infection. And here's an example where there is no nucleic acid involved in the infection, it's just protein. It was difficult to prove that there was no protein in the material that the scientist, his name is Stanley Prusiner, won a Nobel Prize for this. Scientists who worked on this had to prove that it was only protein and not nucleic acid that was causing these infections. And here's some of the evidence that he used to show that it was only protein. So he could treat these samples at 90 degrees C, that's near boiling, for 30 minutes. And that will make nucleic acids fall apart, but it doesn't make prions fall apart. So this is part of the danger of prions, is that people think, well, if I just cook things, I'll get rid of any pathogens in there, and that's not true for prions. They survive. They even survive going through an autoclave, which is high temperature and high pressure. They are small, about 50,000 Daltons. Daltons is a unit of molecular weight. That's the size of a typical protein. Nucleic acids are, like RNA and DNA, are much bigger than this. So they're saying it's not typical of a nucleic acid. RNase is an enzyme that chews up RNA. DNase is an enzyme that chews up DNA. And when you apply these to prions, it doesn't have any effect, so they are unlikely to be RNA or DNA. RNA and DNA fall apart when exposed to UV light, and prions don't. They are resistant to chemical modifications that inactivate nucleic acids. So if you add certain chemical groups to nucleic acids, the nucleic acids can't function as nucleic acids anymore. If you do that same thing to prions, it doesn't affect them. We now know, this is 20 years after Prusiner made his discoveries, that he is indeed right. These are proteins that can act like infectious agents. No membrane around them, just a protein, and they can infect other cells. And how they do it, or how it seems like they do it, is the funny thing about prions is that you have a protein that is a non-prion version of the same sequence in your brains. And you need that protein in order to be a functional human. It folds into a particular shape. The prions have the same amino acid sequence as the protein that you need, but they're folded into a different amino acid shape. So it's sort of like evil Batman versus good Batman. Once you get copies of the bad shape protein inside your cells, they act like a template, like a little factory, to make good copies into bad copies. It can take a while, sometimes like over 10 years after someone is exposed to prions before they start showing symptoms. But what happens is it causes those proteins to unfold, and they unfold in a way that your cell can't deal with them, can't take them apart into amino acids, doesn't know what to do. They wall them off inside a vesicle inside the cell, and eventually the cell dies. So the long name for mad cow disease is called bovine for cow, spongiform for sponge-like, just like SpongeBob, and the last word is encephalopathy. So that just means swelling of the brain. So swelling of the brain like a sponge, turning your brain into a sponge. And people who die from mad cow disease have holes in their brain, and it's from those cells dying over time as they accumulate these proteins that can't be taken apart into amino acids anymore. Okay, so there are other things out there that are not nucleic acid based that sort of act like viruses. But getting back to the question of, are viruses alive? If you take the definitions of cell theory, one of the big ideas of this book is all cells come from other cells. And if we just read what we think is true in what we know to be true according to cell theory and substitute the word virus. So first, for cells, all cells come from pre-existing cells. Cells maintain internal environments that differ from their external environments. Cell structure defines cell function, and cells communicate with each other. So we've seen examples of all that for cells. If we just substitute the word viruses, viruses come from pre-existing viruses as far as we know. That is true. Viruses maintain internal different environments differ than their external environments. Whether or not this is true, this is really hard to measure. Like if the pH is different inside a virus versus outside a virus, that would be an example of maintaining an environment different from their external environment. I don't think there's as good evidence for that. This is another way of saying this is homeostasis, that cells are regulating their environment. Certainly viruses don't do it to the great an extent as cells do. They don't have proteins in their membranes, if they happen to be membrane-coated viruses, that are regulating, say, ion composition or something like that. Virus structure defines virus function. That's absolutely true. You see that giant mimivirus that has got the starfish sort of shape that comes apart to release its genome. Bacteriophage that we saw earlier in the class, that have that little syringe-like sort of structure to inject their DNA. So that's certainly true for viruses. And viruses communicate with each other. Well, in the case of one virus infecting another virus, it is communicating with the other virus and saying, I'm going to use your replication machinery and I'm going to replicate myself instead of you. So are they alive or not? You can certainly make your argument either way. When immunologists talk about making a vaccine, they talk about live or dead virus. Some of the vaccines that you may have taken involve a dead virus. So what that means for a immunologist is a virus that has been inactivated so it can't reproduce. Usually that's with heat. So if it's inactivated so it can't reproduce, then we consider that dead. If you can have dead viruses, then you can have live viruses. But they are not alive in the way that we think of cells being alive, in that they don't have active homeostasis. So it's another one of these complicated topics, in that it depends on how what you consider alive and how you define alive. This guy, Richard Dawkins, is a scientist in the UK, and he's had many influential ideas about evolution. Probably his most famous book is called The Selfish Gene, where he advances the idea that our DNA is a parasite, and we are just carrying it around. So we are just bags of DNA, and our DNA evolved us as organisms just as a mechanism for replicating itself, getting itself into the next generation. This is sort of a crazy idea, but it's a pretty useful idea in terms of thinking about viruses. So if you think about, well, is this the only way to get your DNA into the next generation? Well, viruses are a pretty good mechanism for getting its DNA into the next generation. Certainly, the coronavirus is being really successful at getting it's replicating its DNA. We've gone from just a few cases in December to now worldwide, literally millions of cases. And that's a very short time period to replicate your DNA and a successful strategy. If you're looking at just the point of, I want to get more of my DNA out there. So I just wanted you to know about this idea from Dawkins.